Hi, today we are going to be talking about the Central Dogma. So the definition that you find on Google is the Central Dogma describes a two-step process, which is transcription and translation, by which the information in genes flows into proteins from DNA to RNA to protein. So I'm going to break down how this works. So in a prokaryotic cell, transcription and translation are coupled. Translation begins while the mRNA is still being synthesized. Transcription occurs in the nucleus, and translation occurs in the cytoplasm. Transcription and translation are spatially and temporarily separated in the eukaryotic cells. Transcription occurs in the nucleus to produce a pre-mRNA molecule. Pre-mRNA is typically processed to produce the mature mRNA, which exists in the nucleus and is translated in the cytoplasm. A protein-coding gene consists of a promoted, followed by the coding sequence for the protein, and then a terminator. The protein is a base pair sequence that specifies where transcription begins. The coding sequence is a base pair sequence that includes coding information for the polypeptide chain specified by the gene. The terminator is a sequence that specifies the end of the mRNA transcript. RNA synthesis involves the separation of the DNA strands and the synthesis of an RNA molecule from the 5 cm to 3 cm direction by the RNA polymerase using one of the DNA strands as a template. In contemporary base pairing, A, T, G, and C on the template DNA stand specifically U, A, C, and G, representatively on the RNA strand being synthesized. Transcription begins at the promoter, then proceeds through the coding region and ends at the terminator. The sequence of a prokaryotic protein-coding gene is collinear with the translated mRNA. The sequence of a eukaryotic protein-coding gene is typically not collinear with the translated mRNA. Most eukaryotic protein-coding genes contain sequences called introns, which break up the amino acids coding sequence into segments called exons. The transcript of these genes is pre-mRNA. The pre-mRNA is processed in the nucleus to remove introns and splice the extrons together into translatable mRNA. Eukaryotic pre-mRNAs typically include introns. Introns are removed by RNA processing, in which the intron is looped out and cut away from the exons by snRNPs. The exons are spliced together to produce and translate the mRNA. The process is as follows. The intron is looped out as snRNPs, which also means small nuclear ribonucleic protein, bind to form the spliceosomes. The intron is excised, and the exons are then spliced together, resulting in mature mRNA that may exit the nucleus and be translated in the cytoplasm. And that's what you need to know about the Central Dogma.